So in chapter 16, we're working on proteins. Proteins pretty much are the reason that we're alive. They run pretty much everything in our body, along with maybe some cofactors, some vitamins, and the instruction booklet to make more proteins. So in order for us to live, we've got to have proteins, and we have to have the right number of certain proteins to be able to survive. In the body, the proteins that are made up of amino acids. It's basically like a protein is a long string of amino acids that are held together with amide bonds called peptide bonds. There's 20 different types of amino acids, and we're going to go through the individual generic form of what an amino acid is. But along each one of the amino acids on a carbon, there is a functional group that defines what type of characteristic that amino acid can offer. Whether it's acidic or basic or polar or nonpolar, those are some of the characteristics that amino acids can offer to that portion of the peptide chain. And so depending on how we place the amino acids within the chain and how the actual protein folds together, that is what will determine what the function is. And the functions are incredibly diverse, everywhere from providing structure to transporting molecules around in the body. They increase or decrease chemical reactions. They act as catalysts. They are involved with controlling immunology in our body, and we even utilize some proteins for sources of energy. So in table 16.1, you might want to check that out. That has the different classifications of proteins and what their functions could be, with an example of what actual protein it could be. For instance, with structural proteins, there are two common structural proteins that we find in our bodies are collagen and keratin. Okay, collagen, of course, is something that is within the tendons and the cartilage in our body, and keratin is something that is a protein involved with hair, skin, wool, nails, things like that. So you can stop the tape here and look through the chart. You'll have questions about this on your homework. You may have questions that are basically multiple choice questions that concern this chart. So it's a good thing to know this chart for the final exam as well. Amino acids are the monomer that makes up the polymer that eventually makes the protein. Okay, so they are the monomer, meaning mono meaning one and mer meaning unit. So there's one unit of amino acids that string, each one unit strings upon the other string with a peptide bond. The generalized structure of an amino acid is that it has a carboxylic acid group on one side and an amine group on the other side. And then the carbon that's between the carboxylic acid and the amine will contain what we call the side group, or they also term it the R group. And it's the R group that actually defines what the actual characteristics of that amino acid, what the characteristics are. When we find amino acids, we find that within different pHs, the amine will either protonate and cause it to be an ammonium charged portion of the amino acid and be ionic, so to speak. Or in other environments, it could be that that there's a deprotonation of the carboxylic acid portion of the amino acid, and in that case it will be charged as well, but negatively charged. So when we have an ammonium situation where everything's protonated on the amino acid, meaning we added hydrogens to the amino acid, then it will be positively charged, or called an ammonium kind of version of an amino acid. When something is in its negative form, it means it's been deprotonated. You have an uncharged amine and a carboxylate ion on there. So this is just showing the Zwitter ion form, that you can have a positively charged ammonium on one side and a negatively charged carboxylic acid on the other side. In general, amino acids are chiral. There's one amino acid that's not considered chiral. That is the carbon that sits between the amine and the carboxylic acid has 4 different types of groups around the carbon. The only one that's not is glycine, and that is just due to the fact that it has hydrogens, 2 hydrogens on it rather than another type of R group. So when you have Fischer projections, they're often drawn for, let me put that another way. Fischer projections are often drawn for amino acids to indicate what type of stereoisomers you might be looking at. In general, when we look at an amino acid, if the amino acid has been produced in nature, we'll find it in what we call the L configuration, meaning that the stereochemistry is that is L rather than D. Remember, in sugars, the more natural form of a sugar was a D configuration in Fischer projections, and when we look at a protein, again, it is in the L configuration. So if you were to go buy an amino acid because you were deficient, let's say that you're a vegetarian, so you don't eat meats a lot, so you're deficient in lysine. If that's the case, then you would want to go out and purchase the L-lysine version of it, not one that's a mixed of those isomers, okay? Because that's the one that's going to function properly in the body, okay? So this is just showing you the different stereoisomers of alanine and cysteine, okay? And when you look at the side chain there, that's what's differentiating sort of the difference between one amino acid to the other. You look at that central carbon is the central carbon between the nitrogen and the carboxylic acid is the chiral carbon, and it's the side chain that will determine whether it's the L or the D configuration. Okay. The types of amino acids, again, are classified into different characteristics. We call them nonpolar or polar. Nonpolar, of course, means that it's hydrophobic, and it basically repulses aqueous or water-like environments, and it tries to stay away from there. So if you had a long string of hydrophobic amino acids along your protein chain, those would probably group together and make sort of a globular-like shape where they try to sort of encase other types of amino acids around them that are more formidable for an aqueous environment. So they would glob together. In fact, globular proteins kind of get that name because the hydrophobic core of globular protein is usually nonpolar to try to keep away from the aqueous environment that we see in a cellular environment. A polar, on the other hand, will have groups on the R portion of the amino acid that have an uneven distribution of electrons, causing it to be positive on one side and negative on the other. And so when we look here, we have a nonpolar valine. You can see that the valine is an isopropyl group there, highlighted in yellow, and it's all hydrocarbon. Okay, it doesn't have any polarity because hydrogen, the difference in electronegativity between hydrogen and carbon is not a lot. When we look at the other example, asparagine, this one has an amide on the third carbon removed from the chiral carbon there, and that is a very polar portion of a molecule, and so it would be able to interact with polar solvents, polar environments, let's say. Okay, so the nonpolar amino acids include glycine, alanine, valine, leucine, isoleucine, phenylalanine, methionine, proline, and tryptophan. Methionine, by the way, is also one that usually is derived from meat, so you need to make sure if you're a vegetarian that you get enough methionine in your diet from particular types of fruits, vegetables, and other things that you might ingest when you're a vegetarian. Polar amino acids, on the other hand, will include serine, threonine, tyrosine, cysteine, asparagine, and glutamine, and all of those groups you'll notice have usually, or all of them have oxygens except for cysteine, which is a thiol. Many of them have alcohols. The other two types of R groups that are considered polar would be the two amide R groups with asparagine and glutamine. We also have acidic and basic amino acids, so those would be able to interact and neutralize other acids or bases that they came in contact with, or act as an acid or a base, and depending on what environment they're in. So those are also important amino acids to know. Two examples of, an example of an acidic acid is aspartic acid. Notice that it has that carboxylate listed there on the R group portion. And a basic would be histidine. Notice the 5-membered ring with 2 amines integrated into the ring. We know now from the last chapter that amines tend to be basic in nature as opposed to acidic. Okay, so the 2 types of acidic groups are aspartic acid and glutamic acid. Notice that those were also in the polar. They are polar and acidic. Okay. Our basic amino acids will include histidine, lysine, and arginine. Okay. And again, look at those. All of the basic groups will have that amine on the molecule somewhere within the R group. Okay. So what are you going to have to know for the final exam for this? You're going to need to know how to recognize if something's polar, nonpolar, acidic, or basic. So be ready for that. So when we look here, what do we have for our R group? Well, in a glycine, you only have a hydrogen. That would be considered nonpolar, as opposed to threonine that has an alcohol on the R group, and therefore that would be considered a polar molecule. Okay. When we look at the amino acids, we really do need to pay attention to our diet and make sure that we're somehow consuming 10 of the essential amino acids. Anytime from this point forward that I mention the word essential, it means that it is in organic chemistry, biology, and nutrition. This is a term that indicates that it is something that must be eaten by a human in order for us to be able to get it. In other words, we're not able to synthesize in our bodies these particular structures. So we must get it from our diet somehow. So those 10 amino acids I expect you to be aware of and know by the end of the class, and that is arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Notice that there's only 20 amino acids. So half of the amino acids we actually have to be able to consume. The other half of the amino acids we are able to synthesize in our body. So make sure that you check out table 16.3 that include those amino acids that we must get from our diet. Here's just in table 16.4, we have some of the sources that we can get those amino acids from. Notice that when you look at the vegetarian sources of your amino acids, that they're missing some of the amino acids that we will need, and that includes lysine, tryptophan, methionine. Those tend to be, you have to do sort of a combination of different vegetables and fruits and nuts in order to get the correct amount of amino acids in your diet. So make sure if you're a full-fledged vegetarian that you're checking out your diet with a nutritionist. Okay, let's go back to Zwitter ions and the amino acid. We find that the Zwitter ion form, or the charged form of a amino acid, where we have it positively charged on one end and negatively charged on the other end, is predominant if you find an amino acid by itself. It's usually so we call it neutral, so we kind of describe it the way that glycine is described normally, but it is also flitting back and forth between its Zwitter ion form. We also know that there's ionized forms of amino acids in which the carboxylic acid portion of the molecule could be negatively charged, or other forms of that same amino acid where the amine group is positively charged. Okay, we get the positive charge by protonating all of the molecule, and the amine group actually protonates until it has 4 groups around the nitrogen, making it positively charged. We get the acidic form of the Zwitter ion form when we've deprotonated the molecule. So we get it from the acidic amino acids. Okay, So when we have a positive ion form, it means that the actual amino acid is kind of is within a solution that is very acidic. So it will protonate, protonate, protonate until it becomes positively charged. When an amino acid is within a solution that is very basic, it's going to deprotonate, 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 causing the carboxylic acid portion of the molecule to become negatively charged. Okay, so we can have either negative or positive charges. What we find is that there's a certain pi point or sort of sweet spot where we find it in its neutral form, and if we go above the pi, okay, then if we have a pH level that is more acidic than the pi, let's put it that way, then it's going to be in its positive form. If we have a solution that is more basic than the pi, so it has a higher pH, then it will be deprotonated because it will be sort of engulfed in a basic solution. So this is just an example of looking at the Zwitter ion going to the positive form, and that's where you have everything in the Zwitter ion form protonate, and you end up with an ammonium portion of the molecule. Notice that the carboxylate portion of it also protonated. That makes it neutral on that side, so it becomes overall positively charged, and it has a charge value of +1. Okay, this is an example of when you have it in a basic solution that the Zwitter ion form will deprotonate, so that you end up with the carboxylate negative ion on one end, and the amine will be deprotonated to having only 3 things around the nitrogen, and therefore that won't be positively charged, so it will be overall negatively charged. Okay, so again, if we have a low pH, meaning that there's a lot of hydrogen ions floating around, then we will get a positive form of that amino acid. So low pH indicates positive forms of amino acid, especially if it's lower than the pi value that's listed for it. At the pi value, it will be in its Zwitter ionic form, which is a neutral form of the molecule because it's positively charged on one side and negatively charged on the other. By the way, on this slide, that plus sign needs to be shifted over right above the nitrogen. If we were to increase the pH by making it more basic in the solution, we will find that it's above its pi point, and therefore everything will deprotonate, and the carboxylic acid will be deprotonated and therefore negatively charged. The amine will not have 4 things around the nitrogen, but rather only 3, and therefore that portion of the molecule will not have a charge, but overall that would be a negative charge. Okay. All right, so let's talk about how the amino acids actually come together. We understand sort of the characteristics of the amino acids, that they can be, polar, nonpolar, acidic, basic, polar, nonpolar, acidic, or basic. But we haven't talked yet about what happens with the amino acids when it goes from a monomer to a polymer. So when we take the individual units of amino acids, they'll come together, okay, and form what we call a peptide bond by dehydrating a water off of the amine and carboxylic acid portion of the molecule. So it'll come together and produce a peptide bond. So the peptide bond is that bond that is between the carbonyl carbon and the nitrogen. Okay, the bond between the carbonyl carbon and the nitrogen is the peptide bond. If you were to identify where the peptide bond existed, my suggestion is just to go ahead and circle the whole carbonyl group and NH group, and circle that portion of the molecule and describe where the peptide bond is. So this is just a ball and stick depiction of a peptide bond forming, and notice that peptide bond is there right between the carbonyl carbon and the nitrogen. When we name peptides, you're going to go ahead and use the first, you're going to, let's say, drop the last E off of the name of the peptide, or the amino acid rather, and add a YL, okay? and then put it in the order that it's found in the from the N-terminal to the C-terminal. N-terminal means the amino acid side of the protein chain, and the C-terminal means the carboxylic acid side of the protein chain. So in this example, on the portion that is terminally ending with an amine, that one is alanine, okay? And then in the middle we have a glycine, and then we have a serine, okay? So we would call it alanyl glycyl serine for its nomenclature. Of course, you wouldn't do these with very, very extensive polymers of the amino acids, but when you have small ones, you can certainly use that method for nomenclature. So let's talk about the sort of classifications of the parts of the protein. The first classification is called the primary structure of a protein. The primary structure of a protein is just the order in which you're putting the amino acids on the chain. Okay, for instance, here there's an alanine, and then a leucine, and then a cysteine, and a methionine. So when we look at that, that is the primary structure, just the order in which those amino acids are added. When we look at well, these are just interesting facts, I suppose, that we could talk about. When we look at the primary structures of insulin, it has 2 polypeptide chains, so those has 2 primary structures that are linked together with disulfide bonds between, 2 cysteines from 2 alternative chains. So that alpha chain and that beta chain is the primary structure. The order in which those amino acids are linking together is the primary structure of that amino acid. The secondary structure starts to describe the shape that the primary structure takes on due to the characteristics of the amino acids and the order in which they are, okay? So it's more of a 3-dimensional spatial arrangement of the amino acids, okay? So let's say that we had a polar group here and a polar group here, and they attached, we have an amino acid. So we have amino acid, amino acid, amino acid, amino acid, okay? So in this amino acid right here is polar. This is the opposite sort of charge to it, but it's also polar. If those are in the mix and sort of get all of the different amino acids to come in contact with each other, they would be like magnets. They would stick together, and then the rest of the chain would loop around, especially if those were all non-polar groups. So you can see that according to the characteristics of the amino acids, the actual protein will twist and shape into different shapes. The 2 major types of secondary structures that we see in proteins are alpha helixes and beta sheets. Okay, so I expect you to know those 2 types of secondary structures that could be possible. They're held together by hydrogen bonding interactions. Okay, and they tend to, an alpha helix tends to kind of curl around like a ringlet in hair or a telephone cord, the old telephone cords when we actually used telephones that were connected to the walls. The beta sheet is also held together with hydrogen bonds, but these guys are sort of, you have amino acid, amino acid, amino acid, and then it loops around, and then this amino acid is hydrogen bonded or attracted to the other amino acid. Hold on, I have an alarm going off. Hopefully I can finish a good portion of this. We may have to do this in 2 parts, but we'll see how much I can get finished. Okay, so the beta pleated sheets will come together with hydrogen bonding. You can go ahead and stop the tape if you need to slow down a little bit and see what's going on within this slide. Another type of secondary structure is the alpha helix, and that's where we have the alpha helix, the triple helix, and that's where we have the alpha helixes sort of winding around each other. So you've got 3 alpha helixes that form. Okay, so when we are defining sort of what we're looking at, the primary structure will be the sequence of amino acids on a polypeptide chain. So this is just a question that might be asked on the final exam, for instance. if you were to find a corkscrew shape of the hydrogen due to the hydrogen bonding between amino acids, you'd be describing an alpha helix. If you had 3 peptide chains that wove together like a rope, that would be considered a triple helix. And if we had polypeptide chains that were held side by side with hydrogen bonding, that would be a beta sheet. Okay. Tertiary structures are the shape that the actual alpha helixes and beta sheets will turn around and touch each other. So your secondary structures will be your alpha helixes and your beta sheets. But the way that those interact with one another, let's say you have a beta sheet here and then you have an alpha helix here and then a beta sheet here. So that description of where all of the different secondary structures are and the shape of it overall is called the tertiary structure. The tertiary structure is held together with hydrophobic interactions, hydrophilic interactions. Hydrophobic, of course, is where the polar groups are repulsing water. Hydrophilic is where the polar groups are attracting water. There's also can be salt bridges between the different groups. If there's a charged form that's positive on this portion of the molecule and a negative form here, they can form salt bridges between them and loop the rest of the protein around it. Okay. There's also hydrogen bonds that happen between polar groups and disulfide bonds. And that is just, remember when we talked about oxidation of sulfur groups where they make disulfide bonds, that happens between corresponding cysteines or amino acids that contain the thiol group in the amino acid chain. Okay, so what type of tertiary reaction is happening in a disulfide bond? Actually, you know what? I'm going to skip this. Because I don't expect you to know that leucine and valine are hydrophobic unless you're looking at the actual chart. And I will give you on the final exam a list of the structures and the actual amino acids that correspond to those. Okay, quaternary structure is when we have more than one type of tertiary structure that combine together to make a functioning protein. For instance, this is an example of a heme protein, and the heme protein is made up of several different tertiary structures that are put together to make it functional. And those tertiary structures do not have to be identical. You can have very different tertiary structured or protein groups that come together to make one quaternary structure. Sometimes there's only one type. Sometimes the tertiary structure and the quaternary structure are the same thing, but sometimes it's multiple groups that come together. Okay, so just go ahead and check out table 16.6 and learn that chart. You'll need to know that for the final exam. There will be questions about the primary, secondary, tertiary, and quaternary structures of protein. Okay, so again, the primary structure is the order in which the amino acids link up. The secondary structure is the definition is defined by how many alpha helixes and beta sheets there are. And the way that those curl and form around one another and where everything's placed within the structure is called the tertiary structure. The tertiary structure, of course, is defined or influenced by all of those different types of interactions like hydrophobic, hydrophilic, salt bridges, disulfide bonds, and polar hydrogen bond and polar interactions. Of course, the quaternary structure is a combination of everything that's put together into a functional protein. Okay, so you may have a question such as this where you have to identify the level of the protein structure. If we see a beta sheet, we're talking about the secondary structure. If we're talking about the order of the amino acids in a protein, we're talking about the primary structure. If we're talking about a protein with 2 or more peptide chains, okay, we're talking about the quaternary structure. If we're talking about the shape of the globular protein, we're talking about the tertiary structure. Well, it depends. If you're talking about the protein overall, you're probably actually talking about the quaternary structure. I take that back. And with disulfide bonds between the R groups, we're talking about the tertiary structure there that influences them. Okay. And my answers that said tertiary, that would have been one of those iffy questions, because if you were talking about the shape of the globular protein, you might be talking about all of the different protein chains together. So globular proteins will have compact spherical shapes. They tend to carry out synthesis, transport molecules around in the cell. They are involved usually with metabolism as well. They help with storage and transporting. All kinds of different globular proteins are present in our body. Globular proteins tend to be made up of a lot of alpha helixes. They tend to group or glob in the middle due to their hydrophobic interactions and put hydrophilic amino acids on the outside. Fibrous proteins, on the other hand, will be long fiber-like shapes where proteins build. Okay, those triple helixes that we discussed earlier would be an example of where fibrous proteins sort of are in their secondary structure. So alpha keratin is an example of a common type of fibrous protein. Oops, we skipped to catalyst here. Hold on here. Oh goodness, I'm missing some of my proteins. That's okay. We'll skip to enzymes now. Enzymes are also a type of protein. Enzymes act as catalysts in biological reactions. Remember that catalysts have the ability to be able to lower the activation energy in order to get a reaction to go forward. Without enzymes, we would not be alive because the biological reactions it takes too much energy to be able to get a biological reaction to go forward without an enzyme being present. The beauty of an enzyme, of course, because it's a catalyst, it both causes the activation energy to be lower, but it also regenerates itself after the reaction is over so that we can use those enzymes or catalysts over and over and over again. There is a point where enzymes can be saturated with whatever they're reacting with, but for the most part, if the biology is going along well, then it will just regenerate itself and then move on to the next reaction. But you can set the concentration of enzymes. When you name an enzyme, typically we'll see that the enzymes will end with ASE. So if you hear the word sucrase, for instance, it is an enzyme that catalyzes the reaction with sucrose. Oxidases actually help catalyze oxidation reactions, and enzymes such as pepsin and trypsin are involved with digestion. Let me just check the time. We may have to continue with, we're going to have to continue with enzymes in the next portion of the video. So let me escape out of here. We'll start up with enzymes in the second portion.